Hello everybody, and welcome to this webinar for SOLAS for missing data analysis, especially for multiple imputation in the clinical trials. I'm just a couple of minutes after there, I'm just waiting. There were some people still logging in, so I hope we're all here now. My name is Andrew Grannell. I'm a statistician here at Statistical Solutions. I also have my colleague here with me, Eoghan Murphy, and he'll be helping out with any questions that you guys have throughout the course of the webinar. This should be approximately 1 hour long. I'll be having breaks as we go along, so if anybody has a question to ask, they can ask it. And also you can send in questions at any time throughout the course, and Eoghan will be handling those. So just to get us started, just an outline of the webinar. I'll go through some background information first, just to set the scene of missing data analysis in clinical trial area. I'll then run through the overall SOLAS software package and its features. I'll explain then how through SOLAS you can do your exploratory missing data analysis, looking at the missing data pattern, etc. I'll go through the various multiple imputation methods that we have on offer in SOLAS. I'll run through an example or two of running multiple imputation, and then I'll run through an example of doing some post-imputation analysis. I'll also give you a brief introduction to the script language feature that we have in SOLAS, and we'll have some time again at the end for a Q&A session for any questions that you may have. So the format of this webinar, as I said, you can ask questions. Simply just enter the question in the text box provided for you. Also, if you want to zoom in, if the screen is too small for you on the webinar screen, you can, at the bottom right-hand corner, you can select what percentage you want to zoom in on. So if you want to zoom in on something at any period of time throughout the webinar, just click at the bottom right-hand corner and select the percentage. We will be dealing with, myself and Eoghan will be dealing with questions as they come in, and I will also be highlighting some questions. Obviously, I might not have time to answer all of them, but Eoghan will take care of them through the text box. Also at the end, we will be leaving the session open for a while to handle incoming questions at the very end. There's also a forum set up for this webinar for any discussions that you want to talk about. It's set up on www.solasmissingdata.com/forum at the bottom of the screen there. So if you would like to log in there at any time throughout the webinar or after the webinar for any discussions over the next couple of days, if anyone has any points they'd like to make. We'll also be monitoring that quite closely, so we can get back to you on any issues you may have. So I suppose to go into the background information on this, quite recently the FDA in the US asked the National Academy of Science on Statistics to help them investigate methods to deal with the prevention and treatment of missing data in clinical trials. They did up this report titled the Prevention and Treatment of Missing Data in Clinical Trials. There was a panel who dealt with that, and they made several recommendations to the FDA in how to prevent and deal with missing data in missing data analysis in clinical trials. I suppose the first and foremost point they made was that you should be trying to avoid missing data as much as possible. But as we all know, that's an almost impossible task because there's any range of examples that can lead to missing data in your data set. They also recommended to the FDA that single imputation, such as last observation carried forward or baseline observation carried forward, is not recommended in many cases, as it does not deal with the bias, the possible bias that may exist in the data. two recommendations that I highlighted from their report, and I also have a link to this report on the forum I just spoke about a minute ago on solasmissingdata.com/forum. There's links to this report if you haven't seen it. But recommendation number 12 suggests multiple imputation as a very valid approach to dealing with missing data in your data set, and also the correct approaches in how to handle missing data. And also a topic called sensitivity analysis was also quite strongly suggested by the panel. And I'll just read the recommendation 15. It is a short one, so I'll just read it out. It says, sensitivity analysis should be part of primary reporting of findings from clinical trials. Examining sensitivity to the assumptions about missing data mechanism should be a mandatory component of reporting. So this was one of the recommendations that the NAS, the National Academy of Science and Statistics, put forward to the FDA. They now have taken these recommendations on board and are considering their options in how to create new guidelines for how to deal with missing data in clinical trials. Similarly, in Europe, the European Medicines Agency, the EMA, have a report called Guidelines on Missing Data in Confirmatory Clinical Trials. Now, they feel very strongly that ignoring missing data is not acceptable, especially in the cases of intent to treat. They feel it violates strict principles which require measurement and analysis of all patients' outcomes, regardless of protocol adherence. They also stated that missing data is a potential source of bias, and it is very hard to estimate accurately the variability within your data by just simply ignoring missing data in your clinical trial. And they also very strongly recommend multiple imputation as a method to dealing with the analysis of missing data, and sensitivity analysis to validate the analysis using multiple imputation. So with that background material in hand, we'll now run to the overview of SOLAS. Oh, sorry there, guys. We'll now run to the overview of SOLAS, the actual SOLAS for missing data analysis, the software package, and I'll show you the various features on offer in this package. So I'll close out of this. Now, so when you open up SOLAS, this is the first window that you're greeted with, and from here you can open up a new data set or an already pre-existing data set. The file formats that SOLAS recognize are flat file formats such as .dat, .text, and .csv, and we also recognize Excel file formats, and it can read and write. So at the end, when you're finished, you can also write Excel files. Here's the script. There's a script language window, and I can just show you this here. When you click on View Script Language, so here you can type in your own commands, and then when you execute them, they pop out in the log window. Also, when you run any analysis in the software package, which I'll show you in a minute, the script is generated so that you can go back and alter. If you want to change any of the code, you can alter it yourself, make slight changes, and rerun it again. Very easy to use. There's also a 250-page manual on the SOLAS script language, giving various examples and also outlining all the different possible errors you can get when running code. As we all know, we don't get it right the first time. So also you can get the SOLAS manual quite easily through the help file. So firstly, I'd like to show you an example of the exploratory missing data analysis. So when you first get your data set, you want to see what the data looks like, how much missing data do you have, and what does it look like, what's the shape of the missing data, where is it coming from, are there any particular profiles that are giving you back missing data. So I'll run through this data set first. So this is the health data set. We have 4 variables and we have 500 cases. The first variable is age, BMI, hypertension, and the last one is cholesterol. Now, as you can see here, we have quite a bit of missing data, and the first thing you'd like to do is see the shape of this missing data and can anything be done with it. So before you go into any analysis, you can click View, Missing Data Pattern. And I'd also just like to state here that, as you can see, it's quite evident the data sets are in a spreadsheet format and are very easy to enter and alter and all that kind of stuff. You can also, before I move on, you can also do various things with the variables. You can transform variables, you can group them together, you can split them up, you can define distributions, and also you can highlight, if you've got many variables, you can highlight them. You know, you want to use this variable, that variable for your analysis. Instead of having to scroll across all the variables, you can just highlight them out through here. So just going back to the missing data pattern, there's a very handy feature here. If simply you can just pick and drop if you like, or you can click Use All if you want to see all of them. So this is what the missing data pattern looks like, and I'll just show you the legend there. So the white cells are observed and the blue cells are the missing. So as you can see, for case number 1, we have the age, which is a categorical variable, 1, 2, and 3, and we don't have any other information about case 1. And again, so you see we have the age and we don't have anything else. Similarly, for case number 3, we have everything as observed. But because there's 500 cases, and in many cases there are a lot of cases, you don't want to be scrolling down to see the missing data pattern. So we have this feature that you can collapse it down into a summarized table, and simply it's just View and Collapse. So now this big table has been summarized as so. So as you can see on the rows here, on the columns you have the variable, and on the rows we have 72 variables with this particular missing data pattern. So we have their age and nothing else. We have 20 cases where we have the first and third, don't have the fourth, and we do have the second. We have 358 complete cases out of the 500, and so on. So this is a summarized table, quite easy to see very quickly what your missing data looks like. And you can also have the option of looking at the monotone pattern of your data. So this is what the monotone looks like, where red here is the monotone variable or missing data, and the green there is the non-monotone. So what does this mean? Well, a monotone missing data pattern occurs when the variables can be ordered from right to left, such that a variable to the left is at least as observed as all variables to the right. So for example, if variable A is fully observed and variable B is sometimes missing, then A and B form a monotone pattern. Or if A is only missing, if B is also missing, then variable A and variable B form a monotone pattern. If A is sometimes missing and B is observed, and B is sometimes missing and A is observed, there is not a monotone pattern. So that is quite tricky to follow, I will admit. So again, if you have any questions, just send them in and we can deal with them. And also I can discuss it on the forum later too. So this is the missing data pattern feature that we have. And it's quite easy, quite fast. You can get a quick picture of what the missing data looks like in your data set. We also have another nice plotting feature. We call it, again, here we have scatter plots, histograms, box plots, normal probability plots, etc. We have this plot called the margin plot. And essentially, it's a scatter plot, but also extra information given in the margins, or the axes. So I'll show you what this is for. So say, for example, we want to see the missing data in a given relationship between two variables. So I'm going to take BMI and cholesterol, and let's see what the missing data looks like in the relationship between these two variables. So this is what the margin plot looks like. As you can see, the blue dots here in the main graph are the observed cases, and this looks like a scatter plot. But we have all this extra information given in the X and Y axes. So what these are firstly the red dots down here signify missing data, where we have the observed cholesterol value on the x-axis, but we are missing the corresponding BMI value on the y-axis. And similarly, these red dots here signify that we have the BMI value, but we're missing the corresponding value for cholesterol. And the nice feature here is that these two box plots here below show you the distribution of the observed cases and the distribution of the missing cases. So you can see the distribution goes from about 178 up to about 249, the majority of the data for missing, and the box plot for BMI is the distribution of the missing data looks like this. And the reason why we give the box plot here is because several missing cases could be on top of each other, and you won't know what the distribution of the missing data looks like. So the box plot aids with that. The blue one is for observed, and the red is for missing. Now, also, if you wanted to break this down by a grouping variable, by a group, we can say, if we want to, we can replot this and just modify the existing plot. So I'm going to see what it looks like for the different age groups, age group 1, 2, and 3. So you just drop age into the grouping variable and click OK. And now we've got 3 different shapes and colors. So you can see the blue circle is for group 1, the orange diamond is for group 2, and the green squares are for group 3. And again, the missing data is shown here in the margin. We can also, instead of plotting them all on the one graph, we can break them up into 3 separate plots. Again, modify current plot, and this time we choose use separate plots for each group. Click OK. And now we've got 3 plots, one for each group, the observed data given in the scatter plot format, and the distribution of the missing data for each variable given in the margins, in the axes. Now you can highlight for a particular age group or a particular characteristic, you can highlight what does the missing data look like for each group very quickly. Visually, it's very easy to do. So now we have an idea of how to explore our missing data very quickly just by looking at visual displays. You can also do descriptive statistics on this kind of stuff. So I think now will be a good time just to take a quick break. If anybody has any questions, just send them in, and myself and Eoghan here will deal with them. So I'm going to take a 2-minute break, and you can send in any questions that you have. I'll be back in 2 minutes. Thank you. Okay guys, I'm just going to get started up there again. There was a few questions came in there. Thank you very much for those questions. Just some to highlight there. Does SOLAS run on a 64-bit machines? Yes, it's both 32-bit and 64-bit capable, and it can handle. It's been tried and tested on some very large data sets, so it can handle large data sets on 64-bit machines. Absolutely, yeah. Are there any good books on multiple imputation that can give me a good grounding for starting off? There sure are. The two that both myself and Eoghan found very useful were the book by Don Rubin, Multiple Imputation for Non-Response in Surveys. I think that's 1987. And Analysis of Incomplete Multivariate Data by Schaefer. And that book was, I think, maybe 87 again. 97. So those two books, Rubin and Schaefer, Multiple Imputation for Non-Response in Surveys and Analysis of Incomplete Data, Multivariate Data, those two books are very helpful. Also, in the manual, we have a lot of material referenced that is very useful and gives a good backing. So finally, somebody wanted me just to run through quickly what the box plots were for in the margin plots. So I'll just do that quickly. So these lines in the axes here, on the x and y axes in the margins, what they do is they show, the box plot shows a distribution. So the blue box plot shows a distribution of the observed, and the red box plot gives an idea of the distribution of the missing data. And similarly for BMI. So one for cholesterol, one for BMI gives the distribution of the observed and the distribution of the missing data for each axis, each variable on the axis. Okay, so again, at any time, guys, just put those questions into the text box. And for anyone who's arrived recently, you can send in a question at any time. Eoghan is here; he'll be answering the questions. And there's also the forum on www.solasmissingdata.com/forum. If you want to sign into that, we can have discussions after the webinar on that forum. So I said the next thing I would go through is the multiple imputation side of things. So that was just the so far we've gone through the background info, overview of SOLAS, and the exploratory missing data analysis. So now I'm going to go through describing the multiple imputation side of things. So I'm going to use a different data set this time. I'm going to use MI trial. Also, you guys, these just to mention, these data sets are the sample data sets that come with SOLAS for example purposes. So this data set is a small data set with 11 variables. You're given the observation number, symptom duration, age, the BMI level at baseline, measured again 1 month after, 2 months after, and 3 months after baseline. And we also have the cholesterol level measured at baseline, 1 month after, 2 months after, and 3 months after baseline. Again, you can see here we've quite a bit of missing data. This time we only have 50 cases, just so, for time reasons, that it doesn't block up for running large imputations, large number of data sets. So I'll just show you quickly the missing data pattern. Again, you can either select and drop, or you can just click use all. So here's what the missing data pattern looks like. In this data set. Again, we can just collapse that down, and you can see here there's one person was missing quite a lot of the trial. And again, you can highlight these people out of the data set if you need to contact them or follow up at any time in the trial to try and chase them down for their information or whatever. Then to look at the monotone pattern, here's the monotone and here's the non-monotone data, missing data. So okay, so when it comes to imputation, there are 9 methods of imputation in total. There are 4 single imputation methods: group means, hot deck, last value carried forward or last observation carried forward, and predicted mean. And we have 5 multiple imputation methods: predictive model based, propensity score, Mahalanobis distance, predictive mean matching, and a combo of propensity, predictive mean matching, and Mahalanobis distance method. Now, this package is a specialized in imputation software package, so we offer the most number of imputation methods, and also we have 2 very new ones that nobody else offers, which is Mahalanobis distance and propensity and the combo method. These 2 methods are not offered by anyone else. And also in the script language, there's quite an opportunity to play around with them and change the settings and things that you can run on your own data. So I'm going to do an example here using Mahalanobis distance method. I suppose actually I'll just describe these methods briefly. I won't go into any heavy technical mathematical stuff right now. If anybody wants to do that, we can go into that later on the forum, or you can email us directly at support@statsol.ie. I'll show you the email at the end of the webinar, or you can send in your questions at any time. So predictive model based runs a regression based on some characteristic variables you've highlighted. It runs a regression model, and then for similar characteristics, it takes the model value and imputes those model values into the missing cases. For propensity scoring, again, you select a certain characteristic variable, such as, in this case, symptom duration or age. So people with similar symptom duration and people with similar ages, if they have similar propensity to have missing data, the ones that have been observed will be imputed into the ones that have been missing. With Mahalanobis distance method, you take the people with, again, similar age and similar symptom duration, and from that missing case, you calculate the distance to every other case in the data set, and you can take the top 10 closest people or cases to you, or the top 10%, and you can randomly select from that pool to impute into the missing value. Predictive mean matching is a similar method to predictive model based, where predictive mean matching runs a regression model again based on your characteristics, and instead of imputing the model value, it will impute the corresponding observed value for that model value. So some people prefer the predictive mean matching method to the predictive model based because you're imputing real data instead of model values, because we know that model values have a certain standard error associated with the value, whereas predictive mean matching, you're imputing real values. And then finally, the propensity predictive mean matching Mahalanobis distance combo method. What this method does is it runs a propensity score method first. It runs a predictive mean matching imputation or model secondly. It takes those results from those techniques, and they become 2 additional variables that are put in as characteristic variables into Mahalanobis distance. And then you run the Mahalanobis distance method on those, including the 2 new variables you just created, and you run a Mahalanobis distance method on them. So again, it's a much more accurate model to run. Now these 2 are quite new, Mahalanobis and the combo method. So I'm going to do an example here of the Mahalanobis distance method. So the first options you have, the first thing you want to do is you have the variables you want to impute, and you have your fixed covariates. Now these fixed covariates are what I just described a minute ago, are the characteristic variables. So what do you want to compare people on, or the cases on? So we're going to say people with similar symptom duration, people with similar age, people with similar baseline values. So baseline BMI and similar baseline cholesterol levels. We want to compare them on these. And then we want to impute all these missing cases, we want to impute all these missing cases. So I select all them, I drop them in here. So here are the variables I want to impute, and here are the variables that I want to set as the characteristic variables. You can also select a grouping variable if you have one, or a center variable. You can also highlight or outline how many imputations you want to do, how many data sets you want to create. In SOLAS, there are a maximum of 50. It used to be, a number of years ago, the recommended used to be 5, but now it seems to be quite acceptable in the literature that 20 is about the accepted number of imputations that you can do, but there are a maximum of 50 in this software package. So we're going to do 20. You can also select the or highlight the monotone and non-monotone data by quite simply by just clicking and dropping. Then this donor pool that I was talking about is the values that you take out that you want to randomly select from. So you can highlight that I want to take the 10, in this case, the 10 closest Mahalanobis distance cases to me, to the missing value, are the top 10%. If I want to select this one, it's the top 10%. Again, I can change it to 20% or whatever. So I'm going to take the top 10 closest people, and I'm going to randomly select from that pool, those top 10. And then finally, the advanced options here, you can set the seed, the tolerance, and the stepping criteria. So this seed is very important because, especially for FDA submission, because when you submit to the FDA, they really want to be able to redo the analysis you've done. So this seed is important. And again, you can also alter the seed in iterations of 1 and run many different randomizations and compare the distribution of those randomizations. So this seed option is very important. So there are all the main features. So now I can click OK and run that. So it'll run away and create all the 20 data sets that I want. So now, just comparing, at the back is the original data set, and now at the front I have the new data sets. So right here I've got the red values are the monotone that I described earlier, and the green values are the non-monotone that have been imputed. So just to compare there, for case 7, all we have is the baseline values for each, and this particular case didn't show up for the rest. So now I've got these values entered in for case number 7, imputed in. Also, you'll notice that at the very bottom of the spreadsheet, there are different tabs. So this is the first data set we created. Here's the second data set, third data set, fourth data set. And again, if you just look at the observations here, for case number 7, BMI 1, 192, 258, 222, 258 again. So there's quite a range of values entered in here. Now, these are the 20 data sets. I'm on data set 11 right now. Again, as I did before, if you want to see the relationship between two variables, so I'm going to pick a scatter plot between BMI 2 months after baseline and the baseline BMI value. Select OK. So now here for data set 11, the blue triangles are the observed data and the red are the imputed values. So now that's just for data set 11. If I want to see the distribution across all the other 19 data sets, simply go to View, Lines, View, Imputation, and Show All Points. Now it overlays all the other 19 values that have been imputed on top of data set 11. So this is a quite nice feature because now you can see the spread of the imputed values at each stage over the data set. So that's just an easy, nice task you can do to just explore how the imputation went. If you weren't happy with that method of imputation, you can go back and use a different method of imputation. Also now, you can do analysis on your imputed data sets using the analysis menu. So we can select descriptive statistics, we can run regression models, ANOVAs, and non-parametric tests. So I'm just going to do an example here of some post-imputation analysis. I'm going to do a multiple regression, and I'm going to do it on BMI 2, for example. So when you select that, this output window pops up, and you're given two windows within this window. Now I think this is a very nice feature. We have two windows, variables in the model and variables not in the model. So at the moment, we have nothing in the model. Here's our model. We have the intercept. We also are given statistics like the multiple R, the R squared value, and the adjusted R squared, etc. Again, you can also add other statistics, and also some you can do some run some diagnostics, look at the normality, look at the independence, etc. So at the moment here, and this acts like a stepwise regression. You can put in one variable at a time, and it will recompute. So if I take the most significant, which is BMI 1, which is no surprise, you just click it and drop it in. So there's our first variable. And again, BMI 3 months after baseline. That's quite significant, so I'm going to pick that and drop it in. And now you can see here we're up to 91% significance, or 91% R squared, sorry. And we have a regression equation of the intercept, x1 and x2. Now this is, again, if you look down at the very bottom, this is for dataset 1, page 1. We also have 20 other tabs for all the other data sets. So again, here's dataset 2, models in the model, variables in the model, variables not in the model. And again, you can alter that as well. So here is all 20 data sets. And that's great, but you don't want to be having to redo the model for every 20 data set, or even 50 data sets, because that's quite time consuming. So at the end, what we have is a combine tab that shows you the combined results. Firstly, for all the models, it gives you the, or all the data sets, it gives you the multiple correlation to see how correlated these data sets are. And you can see that it's quite correlated. And it also gives you the mean coefficient value and its associated standard error. So as you can see here, the standard error is quite small in relation to the coefficient, so these are quite nice estimates. So these are, again, quite a handy feature here to get a good understanding of the model. And also, it's important, just to make this point, it's important you don't just select one of the imputed data sets and run all your analysis. You should run all your analysis on all the data sets to see the distribution, or see how see the variance of your results. So it's important to get the standard error of your model estimates to see how accurate the model is across all your data sets. And as I said at the very beginning, that both the National Academy of Science and Statistics were recommending to the FDA and the European Medicines Agency were both recommending that sensitivity analysis be incorporated into your analysis. So sensitivity analysis would include running using many types of imputation and running the same model on the imputed data sets. So we did Mahalanobis distance here, and we ran a model. I can go back and now I want to do a predictive mean matching model and run a multiple regression again on that. So this software package allows you to very quickly run imputations and very quickly run models and just see how the variance and the standard error is incorporated, and you can get a good idea of what methods you want to use and present. Also, what I can show you here is the script that I mentioned at the start is the script language. So if you go view script, up will pop the script language for this data set, or for this method. The first line is just the data set where you read it from. The second line is the variables that you want to impute. Again, it's all commented here. The third section here are the characteristic variables, or the fixed covariates. And then you have all your preferences and what methods you want to use. So if you want to run the code again, you can set up an iteration to use so you can change the seed number, run it again, change the seed number again, run it again. You can also change the method from Mahalanobis to predictive mean matching and run it again and run it again. And you can also select regression, run it again. So you can do all this in the code, but the handy thing is you don't have to write the code from scratch. You can just do all the clicking first to get your starting point, and then you can do all the altering afterwards and the playing around with the code. It's quite a versatile language, and there's a lot you can do with it and a lot you can code up. So I think that just about secures everything that I wanted to cover. So we'd be happy to deal with any questions that you guys might have. If you want to send in some questions, I'd be happy to deal with them. Owen, are there any questions coming in there? Okay, there was one good question that came in there about missing data, and it was, is it always necessary to use multiple imputation, and what is the thresholds? When should you use it? When shouldn't you use it? If you have very little data, should you use multiple imputation? Very little observed data, that is. And the answer is you can run an imputation, multi-imputation, with virtually no data. But there's no point in doing that because we would essentially be making up the data set then. If you only have observed 10 people and you're imputing for 1,000, then you're making up the data, and you're not getting rid of the bias. You're not dealing with the issue of how come 990 cases are missing out of the 1,000. So there is a bit of discreteness to be used with imputation, and the kind of assumptions need to be adhered to. But both regulatory authorities are very strong in emphasizing that imputation is a very handy tool and a very valid tool to be used when dealing with missing data. But a lot of checking needs to be done. As I said, with the sensitivity analysis, you need to run more than one method of imputation, and you also need to compare that back against the same model that you run on the original data set with the missing data. So there's a lot of comparison needs to be done, and you need to see how the estimation of the variability happens, how the variability changes, how it gets larger or smaller when you do imputation compared to when you didn't do it. So, or you could even run a complete case analysis and compare it to your imputation analysis, and see if you find out any more information there. But you do have to be careful with it. But when dealing with imputation, it is quite strongly advised to use multiple imputation when dealing with missing data. Also, there was a question on the graphs. You can save these graphs and read them into to a variety of software packages. You can if you want to put them into a report, you can just copy them to clipboard and paste them into a report, etc. Quite easily. All this stuff is very easy to do. So so I think we covered a lot there. So I'll just put up the final slide there. If you guys have any further questions or anything, you can just put them through the text box that you have there in the webinar window, and we'll keep dealing with them. We'll leave the session open for a while so you can keep putting questions through there. Also, you can contact us directly on support@statsol.ie, and we have this discussion forum especially set up on www.solasmissingdata.com/forum. You can log into that, and we can we can have a back and forth discussion on any topic that's been discussed here today. I sincerely hope this webinar has been helpful to you guys. And again, we're more than happy to help you with any questions you may have on any topic. So thanks again for attending, and we'll talk to you guys again sometime.